This consists of gap 0, gap 1, synthesis, and gap 2, which is the first part of the cell production. Each step has an important function, but the overall job of interphase is to prepare the cell for growth, copy DNA, and prepare it for division. This step is about 90% of the cell cycle.
Gap 0 phase is also known as the resting place. It is a place where the cell has not replicated its DNA nor preparing itself to divide. Some stay there and become differentiated, and some become active again. In Gap 1, cells increase in size and produce RNA and synthesize new proteins and organelles. This step prepares the cell for DNA synthesis, or the S phase. The Gap 1 checkpoint is crucial to the cell cycle because it checks for DNA damage and whether the cell should keep on through the cycle or leave. Within the S or synthesis phase, DNA is replicated. New DNA is synthesized when the chromosomes are replicated. This produces two similar daughter cells where DNA instructions in the cell must be duplicated. In Gap 2, the organelles and molecules required for cell division are produced. The cell will also continue to grow and produce new proteins. The Gap 2 checkpoint is a check for DNA replication and damage. This is the most important because it is the official last stage in interphase before the cell moves on to the last phase, mitosis.
Mitosis is the division of the cell's nucleus. In this step, the focus is to create two daughter cells. This phase is shorter than the interphase and consists of prophase, metaphase, anaphase, and telophase. In prophase, the duplicated chromosomes condense and become visible. The centrioles move to opposite sides of the nucleus and help organize the spindle. A centriole is a small cell organelle seen near the nucleus in the cytoplasm. The centrioles move to the opposite sides of the nucleus and help organize the spindle. The centromere is the round part of the chromosome where the chromosome connects and is the structure of it. Lastly, in prophase, the nucleolus disappears and the nucleus envelope breaks down. During metaphase, the centromeres of the duplicated chromosomes line up across the center of the cell. The spindle fibers connect to the centromere of each chromosome to the two poles of the spindle. Plant cells don't contain a centriole, but animal cells do. During anaphase, the centromeres are pulled apart and the chromatids separate to become individual chromosomes. The chromosomes separate into two groups near the poles of the spindle. During telophase, the chromosomes spread out into a tangle of chromatin. A nuclear envelope reforms around each cluster of chromosomes. The spindle breaks apart and a nucleolus becomes visible in each daughter nucleus. Cytokinesis is the division of the cytoplasm. The process of cytokinesis is different in plant and animal cells. In animal cells, the membrane is drawn until the cytoplasm is pinched into two equal parts. Each part contains its own nucleus and organelles. In plants, the membrane is not flexible enough to draw inward because of the rigid cell wall. Instead, a cell plate forms between the individual nuclei that develops into the cell membranes. A cell wall then forms in between two new membranes.